Hi, I'm Abigail Morris,
And I'm Jessica Smith.
and today we're going to be talking about developmental genetics and immunogenetics in chapter 22 in the book. So our classic paper discussed mutations affecting segment numbers and polarity in the fruit fly, the study of the metameric nature of patterns in the thoracic and abdominal sections of the larva epidermis of fruit flies. Very little is known about any organism about the genes involving the process of spatial organization. Spatial organization is observed when components of biological group are arranged non-randomly in space or on a larger scale, not related to genes or genetics, but think of ripple formations in sand dunes or patterns of waves. There are two basic mutations that are described in this paper. The maternal effect mutations, like where embryos develop two posterior ends arranged in mirror image symmetry and lack a head, thorax, and anterior abdomen. The homeotic mutations change the identity of individual segments. Ultrabiothorax transforms into the metathoracic and abdominal segments into the mesothorax segments. This subdivision of the embryo into segments is thought to occur by a differential response of the zygotic genome and the maternal gradient. Pattern formation. So each gene serves as a different function in the stages of early development in fruit flies. Establishment of the main body axis is the egg polarity gene. Determination of number and polarity of body segments is the segmentation gene. Establishment of the identity of each segment is the homeotic gene. So just continuing on with that, the homeogenic genes determine the identity of individual segments of, in this case, the fruit fly. Mutations in this gene cause bizarre rearrangements of body parts. So our classic paper discussed and looked at several types of these mutations. They're kind of listed below here. So crupple, even skipped, gooseberry, odd skipped, all those are, well, some of those are in your book as well. This is just a table that I pulled from the book. It's on page 670, and it's just segmentation genes and how they control the differentiation of embryos into an individual. They all act sequentially, which means they happen in that order. And if you want to take a look at that, again, it's on page 670 in your book. Okay, so just moving on to chapter 22. So development takes place through cell determination. Each cell in an early embryo stage is totoponent, which means that they have the potential to develop into any cell type. This leads us to the process of cell determination. There were several hypotheses regarding how this process actually worked and how the genetic information might be expressed, which resulted in many experiments with cloning in plants and animals. Every multicellular organism begins life as a unicellular fertilized egg. This single-celled zygote undergoes repeated cell division, eventually producing millions or trillions of cells that ultimately created a complete adult organism. So just moving on to Dolly, I'm sure we all know about Dolly the sheep. She was the first sheep to be successfully cloned. Um, she was, most importantly, the first sheep to be successfully cloned from an adult cell, which no one thought was possible at the time. She's important to research surrounding how cells change during development and whether a specialized cell, such as a skin cell or a brain cell, can be used to make a whole new animal. The photo of Dolly there on the right and her surrogate mother, the reason that they all knew that she was a clone, the first sign was that her white face. If she had been genetically related to her mother, she would have had a black face. So I thought that was pretty interesting.
Okay, so the paper we chose together is called The Immunogenetics of Disease Resistance in Fish, a Comparative Approach. This study was used to prove that the immunogenetic mechanisms that fish carry is anticipated to have an underlying disease-resistant trait. The research included in this study is to show the immunogenetic response involved in disease or immune or health traits can be correlated within genetic differences such as survival challenges and immune responsiveness. So genes in an organism can be influenced by the environment, and survival challenges, including the external world where the organism is located or developed, and the organism's internal world influences factors such as hormones and metabolism. This comes into play with disease resistance because if the organism is in a highly populated population, it will be more prone to diseases being spread, and eventually the disease can go into the body. So the experiment portion of the paper included two approaches to study the genetic influences of genetic resistance. One being selective breeding for changes in immune responsiveness, and another one being a pattern of resistance. Selective breeding was used especially when disease resistance is expected to be under polygenetic control. It focuses on families that were homozygous only for those genes determining the immune response under selection. Pattern of resistance is aimed for a full genetic analysis of host resistance and examined a large number of models, for example, mouse families. In this experiment, inbred lines of species were highly dependent on for success, and inbred lines are basically established by a successful mating of related species for at least 20 or 30 generations. Classical crosses, including F1 hybrids, F2 hybrids, and back crosses, were used to demonstrate Mendelian inheritance. Following the classical crosses, linkage markers were used to map the Mendelian locus on the chromosomes. The methods used to control infectious diseases included hygiene, vaccination, medication, and eradication of infected populations. Also, it has been established that both cold and warm-blooded vertebrates resistance to infection can vary by a wide range of pathogens. Genetics can also play a factor for disease resistance, but the host influences the outcome of immunity. The immune response is regulated by major histocompatibility complex molecules at the surface of antigen-presenting cells, the T receptor, and the surface immunoglobulin molecules on B cells. So this Venn diagram basically shows the overlaps of up and down regulated genes among each fish with bacterial challenges. The numbers indicated up, which is in red, and the down, which is the blue arrow, show the regulated genes in each category. So you can see here that in the diagram how different bacterial challenges affect different types of fish. And this goes into account of all the different development genetics and disease resistance traits these fish can carry. The Biazzi ABH mouse model exhibits a reproducing relapsing disease that accumulates permanent neurological defects. The Biazzi model was used for multiple sclerosis and is proved to show how an autoimmune tolerance eliminates relapses, but fails to stop progression in a model of MS. This is important because this model was mentioned in her paper as the best known example of antibody-mediated immunity by selective breeding. Antibody-mediated immunity involves the action of B cells and secretion of antibodies when presented with a pathogen. When it is exposed to chemicals released by activator helper T cells, the B cell then divides, which produces daughter cells that differentiate into memory B cells and plasma cells. So our selected paper conclusions. The inbred lines of mice and also chickens have proved to identify the response to cellular pathogens when infections come into the body. The immune response in these models have been diverse, caused by different genetic factors, and the genetic factors already mentioned, including population size and genetic transfer. This paper relates the value of immune parameters and its importance of family selection in the disease resistance traits in fish. And these are our references.